So as we start talking about Protista, keep in mind that two of the three domains, bacteria and archaea, have prokaryotic cells. But remember what we said, the archaea actually have some metabolic characteristics that make us think they're more closely related to eukaryotes than to bacteria. So we'll come back and look at some of those characteristics a little bit later, but first we're going to get started on the simplest, in some ways, the simplest types of eukaryotic cells, the Protista. So let's see here. So let's think about Protista in very general terms. Then we're going to come back to prokaryotes for a little bit, talk a little bit more about archaea. So this is a very simple cladogram of living organisms. So remember, a cladogram shows which organisms are related to which other organisms. The more recently that groups branch apart, the more closely related they are. So if we look down at the base of this cladogram, we see a group called Monera. And Monera is a name that was sometimes used for all the prokaryotic organisms. So this is the domain bacteria and the domain archaea combined. Because at least until about 40 or 50 years ago, they were all considered to be the same thing. And then it was recognized that archaea were different enough. They should be placed in their own group. So notice that in this method of classification, which was developed around 1970 by an ecologist named Whitaker, this came to be called the 5 kingdom system of classification, because living organisms were divided into 5 kingdoms: Monera, prokaryotic cells; plants, animals, fungi, and protists. And Whitaker used a couple of characteristics to divide these organisms into his kingdoms. He looked at how they reproduced, whether they reproduced asexually or sexually. He looked at whether they could move. Organisms that can move are called motile, or if you can't move, you're non-motile, if you're anchored in one place like a tree. He looked at how they obtained their nutrients, whether they absorbed their nutrients from the environment, whether they ingested their nutrients, whether they ate their nutrients like people do and other animals, or whether they were photosynthetic. I'm in there. He looked at the number of cells that were in an organism. So whether they were single-celled, which he called unicellular, one cell made up a complete individual, or whether they were multicellular, a lot of cells in one body, in one organism. And finally, the type of cell, whether they had prokaryotic or eukaryotic cells. Now, as we'll see as we look at the 5 kingdoms, a lot of these characteristics, both options, are found in a group. So if we look at bacteria, bacteria reproduce asexually. Different species of bacteria are motile, some are non-motile. So some bacteria can move. Now notice they don't move a lot considering what we might think of as movement for an animal, but bacteria do have flagella. Some of them can kind of glide on their slime capsules on the outside, so they can move. Keep in mind we're talking about very slowly, very short distances, but they can move. They absorb their nutrients. Bacteria cells don't really take a bite out of their nutrients. And that's true if they're living in your cells. So in general, what do we mean if things absorb their nutrients? Well, they either transport individual molecules into the cells, or they release digestive enzymes onto the environment that they're living in They digest the food, but they digest it outside the cell, and then they absorb the food, the nutrients. You've witnessed this if you've ever picked up a piece of moldy bread or moldy fruit, and you've seen how mushy it is. Mold is a fungus, but fungus absorb their nutrients. And there are some bacteria that are photosynthetic. Now, bacteria are unicellular. Each cell can survive on its own. Although keep in mind, there are a lot of bacteria that survive better when they're surrounded by other bacteria of the same species. They sometimes produce non-cellular material that helps them survive. For instance, in your teeth, there's something called plaque that bacteria produce as they're growing. And this isn't really made of cells, but cells make the plaque, and then it can help them to survive. It can help them stay attached to your teeth instead of getting washed down your mouth and digested in your stomach the next time you take a drink of water. Now we're going to skip protists for a minute. We're going to jump up to plants and animals and fungi. So if we look at plants, plants, here it says sexual, but we know now that plants do reproduce asexually. Plants have the ability to reproduce sexually, but they don't always reproduce sexually. For instance, the grass that might be growing in your front yard can produce flowers and reproduce sexually, but you probably don't see flowers coming out of the blades of grass in your front yard in the spring. Grass usually reproduces asexually by sending out runners underground and spreading that way. Plants are non-motile, they can't move. Plants are photosynthetic, except for two weird species. Plants are multicellular, and plants have eukaryotic cells. Now, if we look at animals, again, with all of these groups, when it says sexual, that means they can reproduce sexually. But there are a lot of species of animals that can reproduce asexually. There are some animals that reproduce asexually all spring and all summer, and then in the fall, they reproduce sexually. And later we'll talk about why we might see a seasonal difference. Animals are motile, they can move. Now keep in mind, they can only move at some point during their life cycle. A lot of animals, once they reach adulthood, are stuck in one location. For instance, coral. They ingest their food, they eat their food. Animals are multicellular, and again, they have eukaryotic cells. Now notice, particularly when we're talking about the type of nutrition, how they get their nutrition, when we're talking about whether they're motile or non-motile, whether they reproduce sexually or asexually, remember one of the things you've heard me say before, and that is, it's part of the way that the human mind has evolved. We like to divide things up into nice, neat categories. Nature does not generally cooperate. So there are always exceptions to the rules we're talking about. Now, by definition, all animals, plants are multicellular. All animals, plants, and fungi are eukaryotic, but almost everything else, there's a little bit of wiggle room. Fungi, again, it says they reproduce sexually. Actually, most of the time, fungi reproduce asexually. They reproduce sexually on occasion. Fungi are non-motile, they can move. Although in recent years, we've actually discovered a few fungi that have flagella and can use the flagella to move around, similar to the way that a sperm cell can move by its flagella. And this says that fungi are multicellular. But notice there's a whole group of fungi that are single-celled or unicellular. And that's a group called the yeasts. So the yeast that we bake with or cause yeast infections. or that we use in brewing alcoholic beverages, those are single-celled organisms. Now, when Whitaker was devising his 5-kingdom system, there was a problem. We have a very specific definition for Monera. They have prokaryotic cells. They're the only group with prokaryotic cells. Notice this is a few general characteristics, but when we look at plants, plants have an embryo that develops inside some maternal tissue, something we'll talk about when we discuss plants. Animals develop from a stage after the sperm fertilizes the egg, and this stage is called a blastula. Fungi, at least until a few years ago when we discovered something that were exceptions to that rule, there were no fungi known until a few years ago that had flagella or cilia. And they all had cell walls made of chitin. The problem was when you looked at eukaryotic organisms and you divided them up into plants and animals and fungi, you had some things left over. You had some organisms that weren't plants, they weren't animals, and they weren't fungi, but they had eukaryotic cells. And this is the Protists. The Protists is kind of, was sometimes called a junk kingdom at the beginning. You could think of them as being leftovers. I always think of them as, you know, what you have remaining if you've ever cleaned out your garage or your basement. You know, you put clothes over here, you put toys over here, you put tools over here, and you have another stack that didn't really fit into any of your big categories. It's whatever was left. That's what Protists are. They have eukaryotic cells. That's the only thing that all Protists have in common. Some of them are unicellular, so a single cell makes up their body. The largest Protists are about 30 meters long. That's about 90 feet long. So they're extremely large. Some of them absorb their food. Some of them ingest their food. Some of them photosynthesize to make their own nutrients. Doesn't really say here, but some of them are motile. Some of them are non-motile. They don't really move. Some of them reproduce asexually. Some of them reproduce sexually. So Protists, from the very beginning, it was recognized that Protists were a problem in terms of classification. They had eukaryotic cells, but beyond that, they didn't really fit nicely and neatly into any of the groups. For instance, you might remember from Biology 105, plants contain chlorophyll A and chlorophyll B in their chloroplasts. And there are several groups of Protists that have chloroplasts, but almost everything that photosynthesizes has chlorophyll A. Everything we've discovered so far, but because of what I just said, I don't want to say everything because in another year we may discover something that doesn't. But seems like everything has chlorophyll A, but there are Protists that have different types of chlorophyll or that have even different chemicals. One type of Protist has something called fucoxanthin that it uses to photosynthesize. So when we look at Protists, was realized at the very beginning that this wasn't a very cohesive group. But remember what I've said, we like to divide things into categories. So one of the problems that Protists were a problem at the beginning is they weren't really defined as having any common characteristics. They were defined as being eukaryotes, so they have eukaryotic cells that didn't have the characteristics of plants, animals, fungi, or bacteria. Well, bacteria have prokaryotic cells. Protists are eukaryotic, so they don't have that. Plants have an embryo after the sperm, usually we call it the pollen, fertilizes the egg, and they keep this embryo inside maternal tissue. This is kind of what we're eating when we eat fruits. We're eating this maternal tissue along with the embryo. Fungi don't have undulopodia. That's the technical name for cilia and flagella. Animals develop from a blastula stage. So protists didn't have any of these characteristics, but they didn't have anything that unified them as a group. Now, if we think about protists on our cladogram, notice that protists are shown as evolving from the bacteria, and then different groups of protists evolve into plants or animals or fungi. And that may be true, although we'll talk later about how protists are classified today and kind of update this family tree for living organisms. But when we look at protists, we do know that they probably evolved before plants and animals and fungi. And the reason we know this is looking back in the fossil record. The first organisms that we have fossils of in the fossil record are organisms with prokaryotic cells. And then we start seeing some protists. And we see protists in the fossil record before we see plants or animals or fungi. So we do have evidence that they evolved first, although not by a very long period of time. We had plants and animals and fungi shortly after we had protists. So protists are a problem, and not only because they don't have common characteristics, but when we look at protists, some of them don't really fit well into any group of eukaryotes or prokaryotes. There's a protist called paramecium. And paramecium don't have chloroplasts, but they actually do have single-celled algae that live inside the cells. And these single-celled algae in the paramecium cells photosynthesize. Now notice, this is a good deal for both groups. In nature, the usual rule is that anything that's bigger than you can eat you for lunch. So if you're a small cell living inside a bigger cell, you're protected from a lot of predators. What does the large cell get out of this? Well, since these algae carry out photosynthesis, this means that they give some of those products of photosynthesis, they provide the cell with some glucose. So this is a beneficial arrangement. Arrangements like this are called symbiosis. Organisms live together. This is a particular type of symbiosis called mutualism. Both species benefit. There's are other types of Protista, one called Monocercomanides, that doesn't have mitochondria. Now, it does seem to have some bacteria that live inside the cytoplasm, and these bacteria carry out reactions of aerobic cell respiration. Or if we look at amoeba, the inspiration for the blob in all the science fiction movies, if you've ever seen any of those, it doesn't have a fixed shape. It can change its shape by pushing out these little projections, which are called pseudopodia. Pseudopodia literally means false feet. Pseudo means false or fake. Podia, like a podiatrist, feet. So we have Protista that don't really seem like any other types of living organisms. And so not only do they not fit in with plants, animals, fungi, or bacteria, they don't even necessarily fit in with other Protista. Some of the Protista are very different from one another. However, if we think about this very simple cladogram, Protista in Whittaker's system were thought of as falling into three groups. There were Protista that resembled plants. There were protists that resembled animals. And there were protistans that resembled fungi. So let's think about these three groups. And we're going to come back to cilia and flagella in a moment. So the organisms that resembled plants, the Protista, were referred to as algae. And they were sometimes grouped in what was called the subkingdom algae. So they were in the kingdom Protista, the subkingdom algae. And these were plant-like protistans. They were photosynthetic. Now, if we look at the algae, it includes a number of different groups. The Euglenophyta, notice phyta means plants. There's a group called the Dinoflagellata, which their common name is the fire green algae. The Chrysophyta, the golden brown algae, that's really a romanticized name. They're kind of a dirty brown. I wouldn't call them brown algae. The Phacophita, and this is a typo if you want to correct this on your notes, the Phacophita are the brown algae and the Rhodophyta are red algae. So notice there's fire red algae and just plain red algae. There's golden brown algae and just plain brown algae. And the Phaeophyta and the Rhodophyta are actually two groups that are multicellular. Now, all these organisms carry out photosynthesis, but for one reason or another, they don't have characteristics that we associate with plants. So we'll look at that in more detail later, but usually they either don't have a cell wall made of cellulose or they don't have chlorophyll A and B. There were two main groups of fungus-like protists, and I guess, as evidence that they were insignificant, nobody ever really agreed on what name these two groups should have. But they included a group called the Mycetozoa, which were commonly called the slime molds. And notice if you look and analyze this name, myce means fungus, mycete, and zoa means animal. So these are literally fungus animals. You've probably seen a slime mold if you've ever taken a walk in the park or the woods. You just most likely didn't know you were seeing it because they're so small you might not have noticed them. Slime molds are usually down in the leaves and they kind of look like somebody blew their nose on a leaf or something. And there's another group called the Oomycota. Notice there's that mycete or mycote is fungus, and oo means egg. So literally these organisms are egg fungi, but they were called water molds. And as we'll discuss later, the water molds have been very important because a lot of them cause agricultural diseases in plants. So throughout world history, there have been several occasions when they've had a major impact on human history. And then there was a group called the Protozoa. Now notice proto means beginning or first, and zoa, like in Mycetozoa above, means animals. So the idea is these are the things that evolved into animals. These are the animal-like protists. Now the fungus-like protists made their nutrients by photosynthesis. I'm sorry, the plant-like protists. The fungus-like protists are heterotrophic, but they're heterotrophic by absorption. They don't really eat their food. They land on a surface, they liquefy that surface, and then they slurp up the nutrients. Now, how do they slurp them up? Well, endocytosis. The protozoa are animal-like protists, and they're heterotrophic by ingestion. They eat their food. And they were also considered to be motile, although mostly for a reason that we'll talk about in a second. And there were four main groups of the protozoa: Sarcodina, and Sarcodina means fleshy. There's a type of cancer called a sarcoma, which starts in epithelial tissue, which is the type of tissue in the skin. And Sarcodina moved by pseudopodia. That amoeba was put in Sarcodina. There was another group called Mastigophora, and Mastigophora moved by a flagellum, or some of them moved by more than one flagellum. There was a group called Ciliata, and they used cilia to move. And even though we just said that these guys can move, there was a whole group called the Sporozoa that were parasitic and didn't move. So let's look at a couple of these organisms moving. So I'm going to stop screen sharing here for a moment, go to something else. So let's look at a few of these organisms move. So this is a view under a microscope, and this is paramecium. And paramecium is a single-celled organism. It's a really large cell. In fact, it's such a large cell that it has more than one nucleus. Because remember, the nucleus is where we have the instructions, the DNA for making proteins. If it's a really big cell, it's got to make so many proteins. It's got so many different jobs to do. It needs more than one nucleus. But the surface of paramecium is covered by, depending on the species, hundreds or sometimes thousands of cilia. And the cilia almost look like little hairs. But keep in mind, this whole organism is single-celled. So let's see, hopefully you'll be able to see this movie play and we won't get some screen buffering issues. So you should be able to see the cilia wiggling back and forth. And as paramecium moves, notice that it appears to be kind of rotating. Paramecium has a groove on one side. You can see this groove kind of coming into view. And as it moves, it kind of twirls around. It doesn't always move in one direction. So let me know if the videos aren't clear. And now let's look at an amoeba move. Now again, amoeba is a single-celled organism. So this entire thing is one cell. You can see a lot of little dots in here, a lot of little circles. These little circles are things like organelles that we talked about. So these are things like mitochondria, portions of the Golgi apparatus, things like that. And you're going to be able to see these little particles moving. And what we want to watch is some of these pseudopodia, because what you'll see is amoeba can cause particles in the cytoplasm to flow. And as this happens, it can move towards some stuff and away from other stuff. All these other little things swimming around are even smaller Protista.
Dr. Griffith?
Mm-hmm.
On the videos, you may be clear.
Let me see if I can add some contrast. Unfortunately, there's not a lot of contrast in this one. Yeah, YouTube is not letting me adjust the contrast. That's where I'm borrowing this video from. But I'll try to point out, you might be able to see some movement in here going upward. And you can see a little part of the body coming out over here towards the right. You can see this lengthening. And I'll give you these links so that you can look at them and hopefully see them better on your screen than, going through a couple of screens to get to them. So you can see part of the pseudopodia pushing out over here to the right. I don't know if you can see the particles in there, but hopefully you can see the overall movement as it goes towards the right. Now notice that some of these other small microscopic protista aren't very clear. And this is because when we're looking at something through the microscope, even though it's very small, it's similar to taking a picture with your camera. You probably tried to take a picture at some point and something wasn't in focus, so it looked blurry. And because we're focused on the amoeba, things that are slightly closer to us than the amoeba or slightly further away aren't in very good focus. So we can't see them. Okay, I got it a little bit darker for a moment, not much, but so we'll stop this, but as I say, I'll send you these links and let's look at one more organism. This is an organism that called Euglena, and Euglena moves with a flagellum, but it also has another way of moving. It scrunches up its body kind of like an inchworm. So you'll see its body scrunch up and get real fat, and then it will squeeze and lengthen its body. So here's euglena. You can see the body getting shorter and thicker and then lengthening. Over here, you may be able to tell there are actually two flagella on a euglena. So when we look at these organisms, keep in mind when we say that they can move, they're really moving a matter of inches. So they're not moving very far. So let's go back and look at our cilia and our flagella. So you might remember that when we talked about prokaryotic cells, some of them anyway, have flagella. And it's made of a protein called flagellin. Now, if we look at cilia and flagella in eukaryotic cells, so if we would take, say, a human sperm cell and look at the flagellum, or if we would take that euglena that we just saw the video of a moment ago, or some of the cilia from that paramecium, they all have something that's called a 9+2 structure. And this 9+2 structure is 9 pairs of microtubules around one single pair of microtubules. So the microtubules are made of something called tubulin. So in bacteria, the flagellum protein is something called flagellin with an IN at the end. In eukaryotic cells, the microtubule protein that's in the flagellum and cilia is called tubulin. So here's a flagellum coming out of a cell. So if we look at this, what we see is that this flagellum, and the same thing would be true of a cilia, is really a protein core surrounded by part of the plasma membrane of the cell. And down at the very bottom, there's something called a basal body. And if we look in cells that have more than one flagellum or more than one cilium, what we see is that all the basal bodies are connected by a network of proteins. And these proteins coordinate the movement. Think of it like being in a boat with somebody else. You know, if you've ever been in a canoe with somebody where you're both trying to paddle, if you're trying to paddle the boat forward and the other person is trying to paddle the boat backwards, you don't really make much progress. So in these cells, all the cilia or flagella have to be coordinated, or the cell is not really going to be able to move in a very effective manner. So 9+2 refers to the fact that we have 9 columns of microtubules here, 9 pairs, I should say. So if we count around, we've got 9 of these little kind of teal colored circles represent our 9 columns of microtubules. And then there are 2 in the middle. Now notice the name is kind of inconsistent because we say that the flagellum or the cilium has a 9+2 structure. Well, on the outside, the 9 is 2 pairs, 9 pairs, I'm sorry. But on the inside, the 2 is 1 pair. So it seems to me like they should have named this 18+2 or 9+1. They should have been talking about pairs or single microtubules. But they didn't ask me when they named them. So it's 9+2, 9 pairs around a single central pair. The dynein is another protein. You know, whenever we talk about anything that does anything in a cell, it's pretty much always a protein. This is a protein. Think of something that's dynamic. Dynein is kind of the motor that makes these cilia or flagella. Remember in that video how we saw them kind of waving back and forth? This is what makes them wave back and forth, kind of like the tail on a fish waves back and forth. And that's another difference that we see with a prokaryotic flagellum. In a prokaryotic flagellum, this flagellum twirls around, kind of like a propeller on a plane or a boat. In a eukaryotic flagellum, the flagellum waves back and forth like the tail on a fish. So there's a difference in the protein that makes up the flagellum. There's also a difference in the arrangement of the protein. Prokaryotic flagella don't have this 9+2 arrangement. And there's also a difference in how the flagellum itself moves. Now, they both work to make the cell move. Now, the other thing I want to talk about with prokaryotic organisms is how we get from prokaryotic organisms like bacteria to eukaryotic organisms that have all these internal organelles. And in particular, I want to focus on two organelles, chloroplasts and mitochondria. Because if we look at chloroplasts and mitochondria, we have very good genetic evidence that they both evolved from bacteria. And they evolved by a process called endosymbiosis. So a little while ago, you might recall I said symbiosis refers to organisms living together. Endo, like endocytosis, means inside. So endosymbiosis is living together, living inside something else. And so let's look at a process that we have very good evidence for. In fact, we have so much evidence, this is now called the endosymbiotic theory. And remember, in science, when we call something a theory, that means that we've done a number of experiments that back up this idea, and we're almost positive it's correct. So cell theory, evolutionary theory, endosymbiotic theory, these are all things in biology that we're very sure about. So here we have a eukaryotic cell, this kind of orange colored organism. And the nucleus is represented by this purple oval. And we have a cyanobacterium. Now, cyanobacterium, cyan refers to a blue-green color. Cyanobacteria used to be called the blue-green algae. And so cyanobacterium are photosynthetic. And if we look at a cyanobacterium, it has that plasma membrane on the outside of the cell, but it also has some structures that are very similar to those thylakoids that we saw inside the chloroplast in 105. So suppose we have a heterotrophic eukaryotic cell and it engulfs a cyanobacterium. So it brings it into the cell, and it's going to bring it into the cell by a process called endocytosis. Now think about when this organism gets brought into the cell. When it gets brought into the cell, it's really surrounded by 3 layers of membrane. We have on the very inside, we have that membrane that's similar to the thylakoid membrane in a chloroplast. Around that, we have the actual plasma membrane of the prokaryotic cell, the cyanobacterium. And then around that, we have this little section of the plasma membrane that formed the vesicle that now encases this. So we have 3 layers of membrane. And from what we can tell, one of these 3 layers of membrane was lost over the course of evolutionary time. And so we ended up with a cell containing a chloroplast. And you might remember that one of the things that makes chloroplasts different from most membrane-bound organelles in the cell is that a chloroplast is separated from the cytoplasm by 2 membranes. And when I say that, notice I'm not talking about the 2 layers of phospholipids that make up a membrane. It has 2 membranes. Both of the membranes have 2 layers of phospholipids. Now this process where we have one cell that engulfs another cell, and then over evolutionary time, This other cell loses one of the membranes and becomes integrated into this host cell. This is called primary endocytosis, endosymbiosis. Endocytosis is the process that brings it in. And it's called primary because this means first. Because when we look at protists, what we actually see is that in the case of protists, there appears to have been something called secondary and maybe even tertiary endosymbiosis, where we have another cell that now eats this kind of orangish cell that ate this greenish cell. So we have now 3 layers of membranes around our chloroplast, our photosynthetic organ, because our chloroplast now contains the cyanobacteria, but it also contains the eukaryotic cell that engulfed the chloroplast. And the evidence that we have for this is if we look at the chloroplasts in some of the groups of protists, there are more than 2 layers of membrane around the chloroplast. And in some of them, there's actually a little fraction of DNA, a tiny part of the nucleus that's left. Now notice there also is sometimes some DNA inside that cyanobacteria cell. So when we look, some chloroplasts contain prokaryotic DNA in the cyanobacteria. Some chloroplasts contain some eukaryotic DNA inside whatever's left of the nucleus of this orangish cell. Now, again, if we think of this over long periods of time, gradually most of the DNA gets lost from the chloroplast, and most of the DNA, most of the instructions for the proteins that were either in this prokaryotic cell or the orange eukaryotic cell that engulfed it, most of those end up in the nucleus of the new host cell. Exactly how much depends on which type of organism we're talking about. Now, we have very good evidence that the same thing happened with mitochondria. So when we look at mitochondria, again, remember mitochondria are separated from the cytoplasm by a double membrane rather than a single membrane. And so the evidence is pretty good that they evolved from endosymbiosis. Now, a double membrane tells us a little bit, but by itself, I'm not sure that would be enough evidence to convince biologists that endosymbiosis had occurred. But if we look at mitochondria and chloroplasts, what we see is that they both have prokaryotic ribosomes. So the ribosomes in a prokaryotic cell are a different size and work slightly differently than the ribosomes in a eukaryotic cell. So if we would look inside the chloroplast and inside the mitochondria, if we would look at the ribosomes in there, those ribosomes are actually similar to prokaryotic ribosomes. They're not similar to the ribosomes that are out in your cytoplasm. Now remember, at the ribosome, there's a process called translation that occurs. And translation is when the ribosome reads the messenger RNA and uses that information to make protein. Well, translation can be stopped by some antibiotics. Like there's a whole family of antibiotics called tetracyclines. And tetracyclines affect translation in prokaryotic cells, but they also affect translation in the ribosomes of chloroplasts and mitochondria. So that's additional evidence that these organelles were really evolved from prokaryotic cells. Similarly, if we look at the DNA inside the actual cyanobacteria part of the chloroplast or inside the bacterial part of the mitochondria, that DNA is usually a circular molecule. So remember that bacteria cells have circular molecules of DNA. So when we look at prokaryotic organisms, even some of the ones that have chloroplasts, and we might be tempted to classify with plants, the chloroplasts not only have different pigments, they don't have chlorophyll A and B, but some of them have extra membranes compared to a chloroplast in a eukaryotic cell. But keep in mind that today we talked about the cyanobacteria, which I said at one point were called the blue-green algae. And we talked about protists that used to be classified in a big group called algae. And there are actually some small plants that are referred to as algae. So some biologists are trying to give algae a more specific name. But right now, algae just really refers to something that's photosynthetic. It's usually found in water, so they're aquatic. And it's relatively small. So small photosynthetic aquatic organisms, they're often called algae, but it doesn't necessarily have a very precise biological meaning. So if you read algae, you don't want to necessarily assume they're referring to something that is a protist. Maybe, maybe not. Any questions about protists from this perspective before we leave this page? Okay, if not, I'm going to leave this. Are there any questions about anything we talked about today or anything else? If not, I will let you go. I'll post the links to those videos. But if you just go to YouTube and you type up, you know, cilia or flagella or pseudopodia, you'll have your choice of dozens of videos to look at. And keep in mind, all these organisms that we looked at today are single-celled. We'll talk about some of the multicellular protists tomorrow. And I'm not sure if we'll look at any videos, but we'll look at lots of pictures of protists. So if you don't have any more questions, you can go ahead and go. Don't forget that by tomorrow, I would like you to send me the genus of bacteria that you plan on writing about. Ultimately, that whole exercise is worth 25 points. And choosing your bacteria is only worth one of those points. But to get that 1 point, you have to contact me before class tomorrow. I think so far I've heard from 3 people, but I haven't checked my email since early this morning. So maybe I've heard from more by now. So if you don't have any questions, you're welcome to go. If you have some questions, you're welcome to stay and ask what you would like to ask.
Have a good day.
Bye-bye.
I have a quick question. So, Mr. Simpson, thank you for listening. For the list,
you know, I'm gonna have to tell Mr. Simpson on you, Corey, if you keep calling me that.
I'm sorry, Mr. Griffith.
That's
so
okay.
sorry.
I'm not insulted.
Mr. Griffith, I'm so sorry. You know, I love you. I just had a quick question. Is the list of the different ones that you prefer, is that in the D4L thing? Because I've been having issues trying to navigate that.
Yeah, it's in D2L. It's in the week 4 module, so it was posted at the end of last week. But if you have trouble finding it, send me an email and I'll just send you an attachment.
Yeah, I kind of want to try to see if I can do something around the pandemic, because I just want to kind of learn more about the
well, we'll get a chance to do that later, but keep in mind right now, you know, this pandemic is caused by a virus, so that's a different group that we'll get to later.
Okay. No problem at all. Thank you so much.
You're welcome.
Have a good weekend.
You too.
I'll see you
well, not a weekend, but have a good day until tomorrow.
Until tomorrow. Thank you.
Close enough.
Have a good day.
You too, Jade. Bye-bye.